Hey guys, so intracellular signaling today, basically signal transduction is what we're going to be talking about. So the idea that you can have a receptor on the surface of a cell, which we've talked about membranes and how to put a protein on a membrane surface. Basically, you just send it through the rough ER. But when you do that, that receptor has a very specific structure. And when it grabs onto its ligand, that can initiate some kind of activity. Usually it's phosphorylation. And so a lot of receptors are actually kinases, and kinases are enzymes that add phosphates. They take ATP, take a phosphate off, and will phosphorylate another enzyme. And a lot of times that enzyme is also a kinase. And so what we actually end up seeing is that upstream is what we would typically say is where we have our receptor with its ligand. It's going to grab onto that, and that initiates some kind of action. And so that could either be the receptor itself is now able to shift its structure a little bit, and the domain that can actually phosphorylate is exposed, or it can cause dimerization. So you could go from a really complex, I mean, it's probably a quaternary structure, but let's just say a complex tertiary structure like a full-on protein, and then another full-on protein, and then they can come together and dimerize. And I'm going to show you a picture of what that really means. But oftentimes, what's really happening is this signal transduction is being transmitted through a bunch of molecules. It doesn't have to be a bunch, but it's often a lot of molecules to amplify our message. And we're going to talk about that. The end goal is, like in this chart, it says cellular responses, but really what you're often going for are transcription factors. Either mostly we think of them being like pro-transcription, but they could also be negative transcription factors. And it turns out like there are so many transcription factors that are positive and negative that what the transcription factors that are actually present in the cell are can drastically change how the cell is going to behave, which makes perfect sense, right? I mean, whatever I'm deciding to like, I'm going to make like these 200 proteins versus these 200 proteins, like that's going to change the actual structure of the cell. And that's a way that we can establish, like what kind of cell do you actually want? And so we're going to talk a little bit more about that. The idea of signal transduction, it can be more complicated than just a simple receptor binding its ligand. I mean, you can have, just in this example, you could have one ligand that triggers different pathways. You could have two separate ligands that come together on one pathway. Or it can get real complicated real fast, which is how it actually usually is. But there are some pathways that are less complicated. One of the classic ones would be epidermal growth factor. So it's just one of those things that's going to trigger a cell to basically start going through mitosis. And so you have a classic receptor binding. In this case, you have dimerization, which is a really common theme that we'll actually see in cell receptors. And so you have this dimerization, and it's going to trigger phosphorylation of, in this case, RAF, which is a MAPK kinase, so an MKKK. Oh, yeah, great, KKK. And then that, in turn, phosphorylates another MAPK kinase, which in turn can phosphorylate another MAP kinase. You can kind of see the pattern that's going on here. But they're all just kinases, and they're all just phosphorylating things. And they're glossing over this. Thanks to Khan Academy. They make a really good article on signal transduction. They have really nice pictures. And so you just get that as you continue going on and on and on and on, you're phosphorylating more and more stuff, and eventually you phosphorylate what will become the transcription factor. And the presence of that phosphate allows that transcription factor to enter the nucleus, bind to the DNA, and it will bind to the non-coding region, okay? So that would be like the promoter region or an enhancer region or maybe a suppressor region of a gene. And thus, usually we're thinking about enhancing the transcription, which is going to then, make a messenger RNA, which then we can make into a protein and then express it. But it could be suppressing it as well. An important idea is that each of these kinases is not limited to phosphorylating one of its subsequent molecules or one of its substrates. It's actually able to phosphorylate a bunch. And so let's say, for example, that this RAF phosphorylates 5 MEKs, and then each of those MEKs phosphorylates 5 of these. Well, that's an exponential growth of activation. We call that exponential growth amplification because it's amplifying the actual signal. So you could have the binding of one single molecule, so in this case, the binding of epinephrine or adrenaline to the receptor that's on the surface, and then that's going to trigger a signaling cascade is what we would typically call it. And that signaling cascade is just going to have a series of either phosphorylations or have secondary messengers, which I'm going to talk about in like 2 seconds, and then it's amplifying each time. And so you can end up with one signal receptor being bound, which it's not usually going to be just one, but it could be. And you're going to actually create like 1 million transcription factors that can actually go in and then generate, 100 million of the actual enzyme that's going to do its job. In this case, we're converting glycogen to glucose 1-phosphate. Secondary messengers are a really straightforward concept. If your normal kinase or receptor is phosphorylating something, you can also generate different secondary signaling molecules, and they're a little more nonspecific, so they can go on and then bind to a bunch of different stuff. So in this case, adenyl cyclase is taking ATP and converting it into cyclic AMP, and that could then go on to, in this case, protein kinase A, which is another kinase, so it's going to phosphorylate other things, but cyclic AMP is not specific just for protein kinase A, so it's going to activate a bunch of things. And so that's just one way that you could sort of trigger a variety of pathways as opposed to one direct pathway, whereas a lot of signaling pathways are really, really specific, unlike this. So maybe your adenyl cyclase can only bind to protein kinase A, which can only phosphorylate some other MAB kinase. But in this case, this is actually one that has a broader reach. So I'm an immunologist, and we actually see a ton of this in immunology. So cytokines are protein signaling molecules that cells in your body, especially immune cells, but all cells can use to talk to each other and send each other signals. And one of those pathways is the JAK-STAT pathway. It's one of the early pathways that you actually learn if you take an immunology course, or at least a grad level immunology course. And the JAK-STAT pathway is a series of pathways, I'm going to show you in a sec, that just trigger a variety of different transcription factors, which JAK-STAT is a set of transcription factors that when you trigger them, you phosphorylate them, they dimerize, okay? So you can kind of see they fit together like this little puzzle piece here. That's pretty cool. And so once they do that, they can enter the nucleus and they act as a transcription factor. But you can see here, this is another example of this binding receptor. So lots of cytokines bind to different receptors that trigger different JAKs and STATs. But when they bind together, they dimerize the actual receptor, which allows the receptor to have the correct structure to phosphorylate JAK-STAT. And JAK-STAT is incredibly complex. There's tons of different JAKs, there's tons of different STATs. And so you can see all of these come together. So like if you combine STAT1 with STAT5, that can then come in here and cause T cell proliferation. So if you need to make a bunch of T cells, or maybe you need to trigger your B cells to do something. But depending on which interferon or which other cytokines, so these are interleukins, you get some interferons, those are from viruses. So like if your cell gets infected with a virus, it'll secrete interferon alpha and beta and gamma, and that can tell other cells, hey man, something bad is going on. And so we can look down the list. So we say, okay, I know that interferons are associated with viral infection, and so it's going to trigger these JAKs and these STATs, this is tyrosine kinase too, and then it's going to come down here and trigger those specific ones, which triggers antiviral, inflammation, antimycobacterial, so that's like tuberculosis, and it can trigger those things. Now there are opposite ones too. So in this case, interleukin 10, I just know off the top of my head, is the quintessential suppressor cytokine. And so if your cells are secreting interleukin 10, it tells your body to not become inflamed. As opposed to some of these other ones, you want inflammation because that's going to trigger good immune response. But you don't want that to be too crazy, and so you secrete IL-10. And it triggers still a JAK-STAT pathway, it's just using different ones. And so you can kind of imagine there's a push-pull going on. So what if you had one cell secreting interferon alpha, which is triggering this JAK-STAT pathway, and then another cell nearby is secreting interleukin 10, and that's triggering this JAK-STAT pathway. And so the combination of which transcription factors are actually getting made becomes really, really important. And so going off of that concept, that's a really important idea, and that's going to occur in every single cell in your body. The actual combination of transcription factors changes which proteins get expressed, which is going to change how your cells function and behave. And so in this case, these are all transcription factors. Remember, they don't have to just promote transcription, they could suppress transcription. And so you can have some activators, you could have some repressors. It's really referring to the gene that it's actually binding to, but you can imagine these are all transcription factors binding to promoter sites, or activator sites, or repressor sites, whatever, and the actual combination of them is going to trigger either yes or no, or maybe like a little bit of transcription. And so that combination can be really complicated. Speaking of complicated, let's look at B cells on the left here. These are all transcription factors. It's obviously not complete, but these are all transcription factors, and this is when they're expressed and at what level they're expressed, or what degree they're expressed. Because remember, the concentration of something is also really important, and that's a really important concept to get across, especially in a class like this that's pretty advanced, where it's not yes or no anymore. It's maybe if I have a ton of IRF8, I have no idea what that does. If I have a ton of that, then it's going to actually trigger these precursor B cells. But as that starts to go down and I increase the amount of MIZ1, then I can start going into my pro B cell and then progress slowly. And so you can kind of see there's some patterns that start to emerge. So this blink transcription factor really increases. It's not present in the precursor or the lymphocyte precursors, but it is present, especially as you start going through these different stages of B cell development. And it's actually all one like giant continuous spectrum. And that spectrum we try and subdivide, but you can imagine like how many transcription factors do you actually need in the cell to trigger these different stages? Well, that's a good question. And actually categorizing those is really, really tough. This one over here, I put this diagram on here because you can see it's kind of similar, but it also talks about some of the problems that can occur. And so if you start messing with your transcription factors, that can really affect what your cells are going to do. And therefore, it can affect like the kinds of cancers that you could get. So if you have a leukemic, so like a B-cell leukemia, maybe the Ikaros transcription factor is broken, and so it's not going to work correctly. Or a better version would be like your STAT5 isn't working properly, and therefore you're not able to have your B cells develop right, and now you're not going to be able to make antibodies. That's probably a pretty big deal. The last part of this talk, I'm going to show a 15-minute talk from Dr. Wendell Lim. Dr. Lim is a professor of synthetic biology at the University of California of San Francisco. He's a bigwig there, an awesome PI, and he's going to talk about how they actually use signaling pathways and how they artificially generate signaling pathways. I'm skipping the first half of it. It's about a 30-minute talk, so we're only going to do like the last 15 minutes because he's covering basically all the stuff that I covered already. But he's going to talk about how they kind of approach signaling pathways from a synthetic biology perspective, i.e. like how could you take advantage of this? Like how can we figure out a way to kind of control these pathways and do cool stuff with it? And he's going to spend some time talking about something that's really, really cool and it's super fresh in cancer therapy right now. It's super, super hot, and that's chimeric antigen receptor therapy, or CAR-T therapy. Some of the people that I used to work with when I worked at the University of Pennsylvania, there were some postdocs that were working there. So that means you get your PhD and you just keep working for a couple years to get experience. And they started working at a company that does CAR-T therapy, and they're all making a bunch of money and doing a bunch of really cool stuff. So we're going to cut to him. I'm just going to let him finish it out, so I'm not going to come back. But please listen to this all the way through because you're going to learn a lot.
Another example of looking at the modularity of signaling proteins comes from looking at signaling behaviors in the model cell system of yeast. So yeast is a single-celled organism that has to make many decisions based on what is in its environment. So one of the things that yeast do is they actually have two cell types and they can mate with each other. But to induce that mating response, they have to detect a pheromone that comes from their partner. And so there's a pathway called the mating response that actually detects a mating pheromone, this pathway shown in red, and then that leads to the activation of a set of mating response genes. Now, a completely different pathway that yeast have is a pathway that deals with hyperosmotic stress. So if you put yeast into a high salt environment, it detects that change and then expresses a bunch of genes that help protect it against that osmolarity. So although these pathways are completely different, what does link them is that they're both mediated by a set of kinases known as MAP kinases. The problem is that the cell is filled with many different types of kinases and quite a few of these MAP kinases that are very closely related. And so the question is, if these two pathways, which are physiologically distinct, use shared or similar components, how is it that they actually pass the information, transmit it in the right way, and you don't get kind of crosstalk so you get the wrong response? And so one of the things that was discovered around this time was that there existed these scaffold proteins. For example, there's a scaffold protein that has multiple interaction domains that binds to the mating receptor as well as the set of kinases that are involved in the mating response. They essentially act almost as a breadboard to wire together this circuit so that you get specific transmission of information from the mating receptor to the mating genes. Correspondingly, there's also a scaffold protein associated with the osmotic response pathway, which again binds to that receptor and then also assembles a distinct set of kinases that are involved in transmitting the information to the osmotic response genes. So in other words, both of these pathways can coexist and be specific because there is a scaffold protein that essentially wires them together in a specific way. And so one of the hypotheses that came from this was that perhaps then evolution can create new pathways and by wiring kinases and other catalytic functions together in different ways by generating new chimeric scaffold proteins. So Sang-Hyun Park, a postdoc in the lab at the time, decided to try to test this by asking, could we make a chimeric scaffold protein that would assemble different kinases in these two distinct pathways? And what he found is that when he did this and made the right sort of combination of pathways, of components, he was able actually to make a scaffold that would, when put into a living cell, would now cause the cell to induce the osmolarity response when the cell was stimulated by the mating pheromone. So we could actually create a new input-output pathway, really mimicking evolution. In this case, we had a pathway where the cells would only survive on high osmotic stress when we actually stimulated them with mating pheromones. So really supporting the idea that without changing actually the catalytic components and really only changing the factors that were assembling them, we could create new types of input-output wiring and behavior in the cell. Okay. Here's another example of rewiring that we've been able to do. So Anselm Levskaya recognized that in plants there are these really interesting protein interaction domains that where the interaction is regulated by light. So if you shine a certain wavelength of light, 750 nanometers, on this protein, it will change conformations and now tightly bind to its partner. And so what we realize is that, again, many signaling behaviors in mammalian cells are controlled by induced recruitment. So we asked, could we actually replace many of those interactions? Oftentimes, the catalytic domain will be recruited to an activated receptor, but could we replace that receptor now with this light-responsive protein from plants and recruit catalytic domains, for example, this GEF, to the plasma membrane where it could act on its target GTPase and lead to signaling output? Could we actually control the cell now with light? And so this is an example of linking this light-activated protein to a RAC GEF. So that's going to activate the protein, the RAC GTPase, which itself activates actin polymerization. As you see here, what happens is that when we shine the light now on, which is the red circle, on the cell, you see recruitment of that RAC-GEF and actually protrusion of the cell at that point. So we can actually make the cell follow the light by putting in this novel chimeric protein that combines light-controlled protein interaction with this catalytic activity. So what these experiments hopefully illustrate is that we really can use these sorts of signaling modules to flexibly rewire and reprogram cellular signaling proteins, as well as the networks, and to generate novel cellular behavior. So one of the things that we've become more interested in is asking this second question about applications. Can we actually start harnessing this capability to rewire cells and put it to use? And one of the most exciting things for us is the idea of using this in therapeutics and medicine. That mostly today, when we think about medicine and therapeutics, we think about small molecules or biologics, which are very powerful and wonderful agents. But one of the things that limits them is that they largely will, if you put them into the body, they act in a systemic way. And then also, they largely can just either block or activate something. And they can't actually make very complicated decisions. And that contrasts with, for example, a cell. So a cell of the immune system, for example, which also is essentially a therapeutic, it fights disease, is able to move through the body and find where there's a problem. So it can be spatially targeted. It can also integrate sources of information from that environment and make complex decisions. And it can also execute complex actions like this kind of migration or phagocytosis that we see with the neutrophil. And so one of the visions that we have is if we really understood these principles of how we can rewire cell signaling behaviors, then perhaps we could actually start to customize cells so that they can actually carry out functions that are therapeutic. This would entail, for example, trying to change what sensors the cell has so that they can detect either disease or user-provided inputs. Then we want to also be able to change the cell signaling networks to control how this information is processed and how decisions are made, and then link these to the many interesting and powerful payloads or actions that cells are capable of to really yield now what we consider a much smarter therapeutic response. So one of the great places at testbeds to really explore this today is in the area of adoptive immunotherapy. So immune cells, particularly a type of immune cells known as T lymphocytes or T cells, are an ideal testbed for this kind of therapeutic engineering. These are cells that are able to, for example, recognize and kill a cell that's infected by a virus. So they have very powerful activities. More importantly, we know that we can actually remove T cells from a patient. We can genetically modify them ex vivo. We can expand them ex vivo. And then we can transfer them back into the patient. So this is pretty well established. So we can do a lot of different things that don't have to do with trying to modify things in situ. In addition, T cells can be used for many potential targets. They could be used to recognize cancer. They could be used to recognize chronic infection. And also, they could be used to combat autoimmunity. Okay. So how could we, for example, redirect T cells to recognize cancer? So if you look at a native T cell, the key receptor, a molecule that's used to mediate its action, is the T cell receptor. So if you look closely at that, that has an extracellular domain and then key intracellular signaling motifs. And so, for example, when the cognate peptide antigen is bound by that extracellular domain, it will lead to phosphorylation of these intracellular motifs, which leads to the recruitment of various downstream signaling proteins that then, in turn, lead to the activation of that T cell and, for example, killing of the target cell. What was shown in the 80s by a number of people, including Zelig Eshar, was the idea that you could actually make synthetic or chimeric T cell receptors. So the idea here was, could you take an antibody that recognized a antigen that was only expressed on the surface of a tumor cell? And then, remarkably, they found that if they took this and then added the intracellular portion of the T cell receptor, that this would actually get activated when it recognized a cell with this antigen and lead to T cell activation. So you could actually make these kinds of synthetic receptors, in this case known as chimeric antigen receptors or CARs, that are built of different extracellular recognition domains, things that recognize tumor antigens, but then link together different sorts of signaling, intracellular signaling domains that control the activation of the T cell. And now, 20 years later, this process is refined somewhat, and they're really quite powerful versions of the chimeric antigen receptors that can recognize tumor antigens and stimulate T cell activation. So we have these modular synthetic sensors that can recognize disease antigens and redirect T cell activity towards these cells. So the idea is that we could take these CAR-modified T cells, put them back into the patient, and they should be able to move around the body and seek out those cells that have the cognate antigen and then kill them. So although, again, this seems like science fiction, what we now know in today's world over the last several years is that we actually can use this to attack certain B-cell cancers. This has been remarkably successful in a number of studies shown here, and that there have been quite a few patients that have had a form of B-cell cancer that is non-responsive to chemotherapy that have been cured by using this sort of approach with modified engineered T cells. So this is tremendously exciting, but there are still problems with this. Particularly, what we've shown is that we can take T cells, these powerful cells, and redirect them. But oftentimes there are many strong side effects because these cells are very strong in terms of their immune response. They secrete a lot of molecules called cytokines that really leads to a big systemic immune response. So although we can redirect them, right now, oftentimes they're too strong or poorly controlled. So we really need to learn not only how to redirect this beast, but also how to tame the beast. And so this is where we have been trying to use synthetic biology to generate engineered T cells that show much more controlled behavior. And so one example is the idea that could we actually make T cells that are remote controlled? T cells that only switch on when, for example, a physician provides a drug. We still want these cells to recognize the tumor cells, but we want this secondary switch to be layered over this that allows the user or the physician to control when and to what extent they're activated. And so Chia Wu, a postdoc in the lab, has been doing studies at trying to build switchable chimeric antigen receptors. So this is the conventional chimeric antigen receptor that, of course, has an extracellular antibody that recognizes a tumor antigen, then also has these intracellular signaling motifs. What we decided to do was to ask, could we actually build an additional switch into this receptor by creating a split version of it? So could we take the recognition component, but then take off some of the key signaling modules and put those in a separate molecule, but then bring them together using modules that only heterodimerize when we add a drug? Okay? So the idea here is that the T cell would only get activated when it had this combination of both recognizing the cognate antigen as well as having the drug provided by the user. So fortunately, we've been able to construct a number of different versions of this that work, and one of them is shown here. And so these are some actual assays based on these cells. So what's plotted here is T cell activation monitored by release of a cytokine called IL-2. And so what you see is when we look at the conventional CAR cells, CAR T cells, that if we stimulate with the cognate antigen, which is shown in green, that the cells get activated, and they actually don't care whether or not the drug is present or not, which is indicated by the orange square. In contrast, when we move to this on-switch CAR, T cells bearing this on-switch CAR, we see that they only get activated when there's a combination of both the cognate antigen in green as well as the drug in orange. So here is a case where the physician could actually control when the cell is activated, as well as we've found the timing and the dosage or the potency of that response. What's shown here now is time-lapse microscopy of these switchable CAR T cells attacking tumor cells. So what you see here in the first movie is the T cells are shown in color, and this is one recognizing a cognate tumor cell with the correct antigen. But this isn't the case without drug, and so you see that they interact, but over the course of this 30-minute movie, they don't actually kill that target cell. In contrast, when we now take the same cells and mix them with cancer cells and then add the drug, what we see is this, that we see the cells now engage with the target cells, but then immediately you see this cell blebbing, and then this blue dye is indicative of the cell undergoing apoptosis. So that cell immediately starts dying, and once that cell is on its way to death, this T cell contacts this second cell, reorients its receptors to that, forming a different synapse, and then actually starts killing that. You see the blebbing as well as the uptake of the blue dye. So within this 30-minute period where in the absence of drug, the cell doesn't do anything, doesn't kill anything, we see this, that in the same time when the drug is added, they're able to potently and serially kill multiple target cancer cells. Okay. So we think this is tremendously exciting. We think that in today's world, now that we have the capabilities of engineering and modifying cells, and we understand much more about the principles of how signaling networks are put together, that we're on the verge of really exciting applications where we can use this toolkit of modules to engineer different types of cells that can sense disease signals, environmental or niche signals in the body, as well as user signals, and integrate them to give you customized therapeutic action. And so we think this is a really exciting period for applying synthetic biology to these sorts of mammalian cells, to immune cells, to create exciting and hopefully very useful therapeutic functions.